All right. Good afternoon, everyone. Thank you for joining us for the Environmental Health Language Collaborative webinar. My name is Maria Schatz. I work for the NIHS Office of Data Science. And to remind you that the mission of Environmental Health Language Collaborative is to advance integrative environmental health research by promoting access, use, and harmonization of data through interoperable terminologies and best practices. You can become involved with ELK by learning more about our activities at this web page, NIHS. If you search for NIHS and ELK, you will come to this page. You can check ELK resource catalog, and you can also join our email listserv by emailing either myself or Charles Smith or ICF support at elk@icf.com. Our speaker for today is Sabrina Toro from the University of North Carolina, Chapel Hill, and I will let Chris Duncan, who is a program officer for this project, to introduce Sabrina. Please, Chris.
Thank you, Maria, and thanks to everyone online for joining today. As Maria mentioned, my name's Chris Duncan. I'm a program administrator in the NIHS Division of Extramural Research and Training. And today we're excited to continue our series of ELK webinars focused on highlighting ongoing data and metadata standards efforts in the environmental health sciences. It's my pleasure to introduce our speaker for today, Dr. Sabrina Toro. Dr. Toro is a biomedical scientist and an ontology expert whose work focuses on data standards, biocuration, and knowledge integration across model organism and human disease research. She's an assistant professor in the Department of Genetics at the University of North Carolina, Chapel Hill, and she's a curator with the Translational and Integrative Sciences Lab and also the Monarch Initiative. And she contributes to community-driven efforts in the biomedical knowledge integration and also in data harmonization. Dr. Toro has played a leading role in the development and refinement of major biomedical ontologies, including the Mondo disease ontology, a global framework for disease terminology. Her background also includes extensive model organism curation, particularly in zebrafish, from her time working at the ZFIN or the Zebrafish Information Network. Sabrina is an MPI of the Zebrafish Toxicology Phenotype Atlas Project, also known as ZAPP, which is a community-driven effort to standardize toxicology assays and phenotype reporting and to build a zebrafish phenotype atlas. Today, she'll be speaking about the ZAPP project, which is supported through NIEHS's initiative for accelerating data and metadata standards in environmental health sciences. So with that in mind, let's move along to the presentation. Welcome, Sabrina. Feel free to share your slides and please begin when you're ready.
Great. Thank you so much, Chris, for the introduction. And thank you, the organizer, Chris and Maria, for inviting me to present to you, to talk to you about our project, ZAPP. We are very excited about it. This project is in collaboration with my colleague, Alexa Berger, who is at the University of Colorado, Anschutz. And we're really excited because we're all about data reproducibility. We really want to make sure that the data created by the research community is really usable and reusable, reproducible, and can really help with everything that science should be helping with. This project is motivated by another project that was done at NIEHS. The project is called the CZIT project. The goal for this project was to provide a fundamental knowledge on the use of zebrafish in toxicology and provide scientific basis to decide whether or not zebrafish could be used for toxicological screening. In the context of that project, what they did is to do interlaboratory studies. So they gave the same chemicals to different labs, and they asked them to test them in zebrafish. And a long story short, what they discovered is that there was a big variability between all the results that they would find between the labs. So there was big variability across the design parameters that were between the labs. A big variability of the data that was collected. So what actually assessed in those studies, and also how the data was reported. So we're not really surprised by this variability if you are somebody who has looked at a lot of data coming from the research community. What ZAPP project also showed is that if we ask the labs to use standards to report the data, so in this case, using an ontology term for from the zebrafish phenotype ontology, then we see that the variability between the data being reported is much lower. I would, however, argue that using ontology term is great, but also we also would need to agree on what a very specific term actually means. So in this case, what does an abnormal thermite morphology look like? So the take-home message from this ZAPP project is that reporting phenotypic and toxicology data by using standard would really improve data interoperability, data sharing, data reuse, and data comparison. I think that this is not a big surprise. I think everybody agrees that using standard is a good idea and really should be done. When we talk to the research community, everybody agrees also. We need to use standards, we need to use the same language, but at the same time, the research community does not report the data using standard. And when we talk to the research community, the main reasons are that they don't know what standards to use, they don't know how to use the standards, they don't know where to find them, and then also they don't know how to use standard and integrate this in the workflow of their research, because in their mind, using standard means multiplying the time for the time that they use to do an experiment. It would be multiplying it by at least 2 or 3. So what our team is trying to do and what our project is about is that we want to help the community in using standards. So we want to either develop new standards or integrate the existing standards for phenotypic and toxicology data. We want to make those standard adoption approachable so that the research community would use them. And this project is really community driven. So we are supporting the community to push this project so that everything that is created and done via this project is really meets the needs from the community. So the Zebrafish Toxicology Phenotype Atlas, we're also calling it ZAPP for short. Our ultimate goal is to create a Zebrafish Phenotype Atlas. It would be a web application that has images, and these images would represent a very specific phenotype. So we could have a visual definition of what a phenotype would look like. This atlas could be explored and filtered and searched so that people can find whatever images they're looking for. And every single image would be annotated with all the information that are required to understand how this phenotype came to be. So there would be information about fish, information about the toxicology assay that was done, and what the phenotype is. All of those images, as we just mentioned, will be annotated in the same way and will be available in the atlas following very specific standards. So we want this atlas to have all the information standardized. So we are also working on those standards. So creating the standards when they don't exist and reusing and integrating the one that already exists. Our plan is to have the community, the research community, submitting all the data that will be available in the atlas. The research community already collects the data in the lab. This is already what they do. So we really want to get that data from them. And in order to have that data in the atlas in a format that follows very specific standards, we are creating tools, so annotation tools that will allow the community to annotate the data following standards and using standards so that they can submit to the atlas, and generally so that they can create data that are born interoperable or born standardized, if you want to think about it this way. And because of that, of course, this annotation toolkit will represent all the standards that were created or decided on for toxicology and phenotype. This project is fully driven by the community. So we want to make sure that the community is at the core of the project. As I mentioned, the community will annotate and submit the data, but then also we want to include the community in the standards development, because they really are the knowledge experts. And so we want to make sure that what we create and what we are putting out there really fits what they need and fits, quote unquote, the reality of the data. So the community is also included in the requirement of all the products that we are working on. And also the community is included in all the governance structure that we're creating. So the community will decide how to reuse data, how to attribute data, all of those sort of things. Community can mean a lot of things. So for this project, really the community is whoever wants to be a part of the ZAPP community. Mostly zebrafish researchers, as I mentioned, they are the one in the lab doing the work, but also toxicologists, people who are expert in creating atlases, people who are expert in creating standards. Anybody who wants to be a part of it is invited to really be part of this project. So before we get into the details of each part of this project, I want to start with a few caveats because it helps also put things into context. The first thing is that everything we're developing is for zebrafish and for zebrafish data, but we are also trying to make it so that it can be as species agnostic as possible. So all the tools that we're creating should be, somebody should be able to take them and reuse them for any other toxicology assay. So including the data model, we are making sure that anybody could take what we have and then with little tweaks could use it for other animal models, other toxicology assay. And the reason we are doing that is to make sure that not only the standards are there for zebrafish, but then also that we can compare across species and across models. The second thing had to do with phenotypes, and this phenotypes, I'm confused. This is exactly, it's a quote from somebody in the audience last week when I gave a talk about this project. And the confusion came from the fact that phenotype can mean a lot of things. It can mean the morphology of the animal is abnormal, but it can also mean a change in gene expression. It can mean a change in behavior. And this is true. Phenotypes is a very big concept. So in this case, we're really focusing at this point, first iteration, we're focusing on abnormal morphology. So anything that affects the morphology of the animal. But we are creating that foundation so that if later on we want to add videos, for example, to show a change in behavior, or if we want to show something like change in gene expression, we should be able to reuse what we are already building and just adding this. Similarly, when we talk about fish, it could mean so many things. It can mean just wild type, it can mean mutant, it can be transgenic. For this very first iteration, we're really focusing on wild type, and wild type that were treated by a chemical. So wild type that went through a toxicological assay. Again, everything will be set so that if later on we just want to add simple mutants, everything, the foundations of this atlas will be there, and we can easily, quote unquote, easily add all that information. All right. So I've been talking about standards and the needs of standards. So let's just go into this subject. And what we talk about when we talk about standard in this context is really two different things: the data model and ontologies and standard vocabulary. So a data model is very basic. Very basically, it is the formalization of all the pieces of information that are required to make sure that the data makes sense and to make sure that the data can be reused. And it is not only all the pieces of information, it is also how these pieces of information should be reported. And we'll look at some example in a second. The how this should be reported has to do with this other box here, the ontologies and standard vocabulary that are basically standard language. How do we report a very specific piece of information? So let's look into the data model that we have. So we created a data model for toxicological exposure for zebrafish. This data model was created based on community input, mostly researchers and toxicologists. And then it has been refined based on community feedback and input. And we expect as time goes that there's still some tweaks to be done, but I think we are in a really good starting point. And as I mentioned, this data model should be easily tweaked or reused for any other animal model or toxicological assay. So looking at it at the big picture level, I'm not going to go into details, but if you're very interested, you can come and get more information here. And I think the slides will be shared. So you can have those links, or you can ask me a question later. So basically the way we look at it is that we're representing a fish that was exposed to an exposure event. The fish, if you're doing something else like NAMs or mouse or whatever, you could replace fish by another organism, another model. So it is a way to show that this data model could be used for some other organism than zebrafish. For representing fish, we are leveraging an identifier created by ZFIN. ZFIN is a zebrafish information network. And it's basically the database for zebrafish data. And they already get all the information to do with alleles and variants, and they're already creating those identifiers. So we're leveraging what they're already doing because we don't want to reinvent the wheel, and all those identifiers are already ready to use. So we're leveraging the identifier from ZFAN. Looking at the exposure event, so the fish is exposed to an exposure event. This part of the data model includes a lot of information, including the stressor. So what chemical was, to which chemical the fish was exposed. And for this, we are using the CABI ontology, the chemical ontology, how this chemical was put in contact with the fish. So this is the exposure route. So if the chemical was put in the water, if it was injected, if it was put in the food. For this, we use terms from the exposure ontology. Those terms did not exist. And so we worked with the exposure ontology team, and we created those terms. Similarly, we have information about the regimen. So if the exposure was continuous through time or was repeated. Similarly, we created those terms in the exposure ontology. And finally, all of this exposure leads to a phenotype. And for this phenotype, we are using the Zebrafish phenotype term, which we'll talk about a little bit further in more details. And this is a place that if you're not using Zebrafish, if you want to use a different animal, then you would use another phenotype ontology term here. So this is the high level view of our data model. As I said, if you want to have, if you want to go into more details, we can, but I kept it higher level for this presentation. And so using this data model, then we can have all of those all of that data coming from toxicological assay annotated in a way that is standardized. So this is an example from this publication here. We will have this image of the fish and then information about the fish. So that was an AB fish that was exposed continually with this chemical, and the chemical was put in the water, and the exposure was between this life stage and this life stage, and the phenotype was assessed at the 96-hour post-fertilization stage, and that was what the phenotype observed. So decreased interocular distance, and all of these pieces of information are represented by an identifier. So this is what we think about when we talk about standardizing all data and all data from coming from the research. And as I mentioned before, and I did it here, there's more information. There's more information that one can add here, but these are really like the important information for this context and this discussion. I want to take a little bit of time, not much, to talk about those specific standards that we use to report each of those data type. And for these, we use ontologies, and we reuse all the ontologies and terminologies that already exist. We have been, we being our whole team, we have been there long enough to know that nobody should ever recreate a standard, especially not an ontology. Instead, we need to reuse what already exists, and then work with the team to make sure that we can improve or enhance those ontologies so that we have all the information and all the terms that we need. So in this project, we use the EXO ontology, exposure ontology. I mentioned it earlier. It is to report the exposure route and exposure regimen. This ontology is species not specific, so anybody can use it. And as I mentioned earlier, we needed those terms for route or regimen, and we just created them and added them to this ontology. So now these terms are for everybody to use. We also work with the environment exposure ontology, or ECTO. It is also an ontology that is species non-specific. And right now we use this ontology to express the fact that we're looking at exposure to a chemical. So we use this ECTO term to say the assay was an exposure to a chemical, and then we add the information about the chemical. But also what ECTO will do, it will allow us to represent other type of exposure. So for example, exposure to fungus extract, or exposure to this plant extract, or exposure to smoke. ECTO has those terms, and ECTO will allow us to add this level of exposure. Right now, we really focused on exposure to a chemical because this is where what people do in the lab. So iterative process, right? We're starting with one thing, but we know that we want to expand using ECTO. And lastly, the zebrafish phenotype ontology. This ontology is specific to zebrafish, but it has already been integrated to another phenotype ontology called UPheno. This UPheno is a species non-specific phenotype ontology, so that any data that refers to a zebrafish phenotype ontology term can be integrated and compare more relatively easily with other species, other species, phenotypes from other species. I think that's the way to say it. Yes, specific to zebrafish, but we know that the ZP allows us to compare with other species. ZP, so zebrafish phenotype ontology, ZP, it's a weird ontology, I'm not gonna lie. It was created computationally based on ZFin phenotype annotation. So the way ZFin annotate the phenotype, they do not use an ontology terms. Instead, they use multiple ontology terms. So for example, instead of saying microencephaly, they would use an ontology term for brain. They would use a different ontology term to say decreased size, and then they would say it's abnormal. And this is the way for them to say that the phenotype is a small size brain. And so because everything, this whole ZP was created computationally, I'm sure you can imagine that it is a bit crazy. The names are very non-intuitive. So instead of microencephaly, the name would be something like brain decreased size abnormal. So one of the things that we are doing, and really actively at this point, is to review this phenotype ontology and make sure that the labels are user-friendly. So researcher can more easily find what term they need. And also we are adding definitions. So something, again, we're reusing something that already exists, zebrafish phenotype ontology, but we're also working and enhancing and improving it so that we can all use it. So in summary, for the standard aspect of this project, we have a data model for toxicological exposure for zebrafish. You can learn more about it here. This data model is LinkML based, so also a data model that can be easily integrated and compared with other data model. These are all the ontologies that we are working with at this point. There might be more in the future, but these are really the top ones. So feel free to go to any of these and investigate them. And if you want to use these and you don't find the terms, or if you don't find what you need, this is how you can get you can get to the team and help and participate in the development of those standards. So as I mentioned, we have standards, and now we want the community, the research community who creates the data, we want them to annotate the data so that they can submit it to the atlas. So this next section is all about adoption of the standard that we are creating. And of course, we want researchers to annotate the data so that they can submit it to the atlas. But we're also thinking a little bit further and thinking that researchers should really annotate the data using standard, but then also they could download those annotations and publish them whenever they're publishing their own studies. So we are also really inviting people to download the annotated data and maybe adding them as a supplemental data to the article or the manuscript that they're submitting, because I think that would really help with the research data to really be born standardized if we want. Again, the ultimate goal, the way we look at it, is that the ultimate goal is really to make sure that the data that is reported by the research community is unambiguous, comparable, reproducible, and reusable. And that's really our main goal, is to make sure that the data is standardized. So the way we look at it is that this is our ultimate goal. We know, as I mentioned to you a few minutes ago, that the research community is willing to use standards, but they don't know how and why. So we are really trying to do everything we can so that the community can use standard and report the data using standard without them without them having to understand anything about how standards are developed, how ontologies are developed. Eventually, they will learn based on what we're going to show them. But anybody should be able to come to the annotation tools that we're creating and be able to standardize the data. And as I mentioned earlier, so that once they annotate the data, they can submit it to the atlas, or even download them and submit those annotated data with the submitted manuscript. So how do we do that? So as I mentioned, we're creating an annotation toolkit that looks somewhat like that. This is a picture of a prototype. We make it easier by having the common names as autocomplete. So if we look at the fish section, looking at the wild type, a user can start typing the name of the wild type. Everybody in the zebrafish community knows the AB wild type, or ABTU, or ABTL. And so what we do is that we have an autocomplete with all the possibilities, but we are also showing the identifiers. So people don't need to use the identifiers, but by showing that, they start getting aware that behind the word AB, there is a concept, and there is an identifier. So we're bringing awareness of the whole standard. In other cases, so for example, in the part where users are reporting the exposure route or the exposure regimen, this information in the data model is very constrained to very specific terms. So for zebrafish, in this case, we have very clear ways of exposure routes that are allowed. So we just give those options, and users can pick whatever work for them. We try to make words that are user-friendly, so that the names that are there are names that people understand and people use on a general basis. And then we translate behind the scene. We translate all of that information into identifier. So, for example, if somebody tells us that the exposure route was that the chemical was in the water, what it means, the term that is being used is basically ambient aquatic environment route, which doesn't mean very much things for regular researcher. And the last thing, and really to me very exciting thing about this this project is for the time when somebody needs to pick a phenotype ontology term. So when somebody reports a phenotype, as we mentioned earlier in this presentation, it is very important that they use a phenotype term, and for us, it's a Zebrafish phenotype term. And as I mentioned, the Zebrafish phenotype ontology, first of all, it's an ontology, so it's a bit complicated for a non-ontologist to follow up, but then also it is hard to find the term that you need. So we are creating what we call the ontology term picker that will allow anybody really to find the phenotype without having to understand how ontology works. Again, this is a mock-up of a prototype. The way it's going to work is that if somebody can search using a phenotype term, for example, microencephaly, and then here we would have results for phenotype terms that are called microencephaly, either for the label or synonyms. And then this information here about the term, so the different synonyms and the definition. You can also see here the identifier for this phenotype term. If somebody comes here and look at this and think there's something wrong, for example, here microencephaly is a good name, but it's possible that the name of this term is not something that researchers use on a regular basis, we will have an easy way for people to request changes or to report, let's say, something that needs to be changed, like here. Also, we are developing these tools so that users can look for a term using another anatomy term. So in this case, somebody will look for brain, and by searching for brain, they would get information, like they would have options of phenotype terms that they're referring to something going on with the brain. And then by filtering or adding more information here in the text box, then a user can find the term of interest from this list. So here, by adding increased size, we can see all the different terms that exist that has to do with brain and increased size. Like here. We're also showing the ontology classification here, so that we know that using the more specific term is really helping down the line. It's better to use a more specific term than a higher level term. So by showing that to the users, we're also nudging them to understanding that an ontology has this sort of classification, but we're also nudging them to pick a more specific term if there's one that is useful. And if they cannot find the phenotype for whatever reason, then we also have an easy way for them to report it and ask for a new phenotype. So we are really making it simple as possible for them to find the term, but then for them also to participate in the development of ZAPP by reporting terms that are needed and reporting problems or issues that they find. This term, to our knowledge, there's no tool like this out there that is publicly available. We're really excited. We're really excited about this tool, and it is developed so that it can accommodate any ontologies, especially when you have two concepts that are represented in this ontology. So in here, it would be we're using it to find phenotype terms based on anatomy, but we could use this tool for finding breeds based on taxon or disease based on genes. So this tool can really help with adoption of standards using ontology term by helping people find the terms. So this annotation tool, it's coming soon. We all know what soon means. We don't have a specific time, but we already had a prototype that was tested by a handful of researchers, and we're tweaking all of these. So this is all very exciting. Just quickly, I want to talk about the web application. We don't have anything done yet, but the idea is that anybody can come, anybody can filter, browse, find whatever they need from in this ontology, in this atlas. We are working with the community to get the requirement. So we want to make sure we understand what the community needs, and then we'll make sure that we address those needs as best as we can. We already know that the main feature from this atlas is that it will be open access, so everybody can use it. All the content in this atlas will come from the community and will contain data that is standardized. And we will also have all the content, and all the data will be attributed, and provenance from that data will also be available, because we want to make sure that everybody who contributes to that atlas get credit for their work. There's also a discussion about peer review of the data. If somebody submits an image with a phenotype and everybody else in the community disagree, we're talking about how to handle these situations, and all of these will be discussed with the community. So stay tuned for this. We don't have details yet, but there's a lot of ideas and a lot of discussions about what it could look like. So I want to finish this presentation by talking about community and community involvement. As I mentioned earlier, the community is really a key actor in this whole project. The community is involved with data collection, annotating the data, submitting the data to the atlas. The community is also helping with the development of standards and giving us all the requirements for everything in the project. So requirements for the atlas, but also requirements for the annotation toolkits and how data is submitted. Also, the community is at the core of the discussion about governance. So how is the data going to be accredited and attributed, and how is the data going to be shared? How are we going to review the data? All of that governance is being discussed, and everything will be decided at the community level. And as I mentioned earlier, it is referring to an earlier slide. The community is really with us, and the community is really wanting this, but the community really doesn't know how to do it. So I want to spend the rest of the next 3 minutes basically talking about how we work with the community and how our outreach seemed to really be working, and we've been very successful. And really, I think the key is that we just meet the community where they are, and we help them with whatever they need, and we really nudge them slowly into using standard and talking with us. And eventually, we know that this process will lead to them having a better understanding of standard and a better understanding of how to handle the how to adopt the standards. So we're meeting the community where it is. As I said, we develop all those tools to make sure that the standard can be adopted in an easier way. But we also make sure that we speak their language. So, for example, every time I talk with the researchers, I don't use the word data model because I know that they will not be with me if I use the word data model. Instead of saying, we need your requirement, we really, we have workshops and we have exercises so that we can get the information without having to use words like requirement and have this, this confusion about what we are really talking about and what we really need. Also, we're spending a lot of time fostering the community awareness. So in the Zebrafish community, research community, and I think it's a little bit in all the research community, if you go to a conference, there will almost never be a presentation about standard, or even more, like there will never be a session to talk about standard or even to talk about community projects. So we are really working hard to work with the, not only we present everywhere we can, poster, presentation, we do workshops, but also we work and we partner with conference organizers so that we can request and really push, I would say, to get sessions at this conference so that we have a place for everybody to listen and for everybody to hear about standards and about community projects and about how people can really be involved in those community projects. And I have to say that this is really this can really happen thanks to my MPI on this project, Alexa Berger. She is a zebrafish researcher. She's also on the board of the International Zebrafish Society. So she is at the perfect spot to really make that push and communicate to the research community. And lastly, we really try to do everything we can to really clearly communicate what people can do and how they can participate. It has to be really, really clearly stated. So this is the slide that the summary slide that I show to people. And this includes all of you here. If you want to participate in the project and you want to be part of the ZAPP community, please do. You can sign up here at this link. This is the QR code for signing up. We also have a Slack channel. This is the Zebrafish Rocks Slack. We can give you a link if you're interested. We also have a lot of workshops. So you can come to the workshop, give your feedback, participate in all the activities we do so that we can get information about standard, about requirements, what you expect from this project, and really talk with us and send us feedback. What do you really need? What is it that you want? What can we do to help you in whatever work you're doing? But also we need to know when there's anything that is not correct or anything that needs to be changed or something that is not correct, because really the research community is the place where the knowledge is created and where the knowledge is, and we all need to make sure that whatever we create makes sense and is useful. So this is the summary of this project. Again, our goals are to create or integrate existing standards for phenotype and toxicology data. We are doing whatever we can to make the standard adoption approachable, and we are really fostering and supporting the community into this whole process, because our ultimate goal is to make sure that the data that is created and reported is unambiguous, comparable, reproducible, and reusable. So a big acknowledgement to the whole team. We are two major lab in this project. There's my team at UNC. We are a dry lab, so we're really focused on everything that is standard and computation side of things. And then my colleague Alexa Berger, who is at the University of Colorado Anschutz, who is the wet lab, so really focused in the fish researcher community. We also have a big advisory team that helps us with, this team includes expert in standard development, community-driven project and toxicology data. This is our website. This is where you can send us an email and where you can join our community. And lastly, we want to thank the NIHS for the financial support. And I think there's a little bit of time for questions. Thank you so much for listening.
Thank you very much, Sabrina. Wonderful presentation. This is a good time for the audience to ask questions. You can either raise your hand or put your question in Q&A box. And Laura, please go ahead. Can you unmute yourself? Yes.
Yes, I think so. So
yeah.
Thank you for this effort. I am 100% behind efforts to try and improve reporting of experiments across the board. One thing that I noticed on one of your slides, and it is entirely possible that perhaps it was cropped out or something, but it looked like you only had 2 exposure options, either a what was it? It was a continuous exposure or a repeat measure exposure. How are you capturing static single exposures or pulse? I'm presuming the repeat exposure is for pulsed exposures, but there are several or many studies out there that do a static exposure, and that does not seem to be captured or be capable of being captured on the dashboard currently.
Yes, thank you for this question. You're correct. Right now, what we call the regimen, we have 2 options. So one that is repeated, so whatever exposure and whatever amount of time of the exposure is repeating through time. And the other one is what we call sustained or continuous, that you have the exposure through a certain amount of time. And it's for this one, it could be that the exposure, the solution is maintained the way it is all the time, or it could be changed. We have those only 2 options because reviewing the Zebrafish experiment, this is the 2 most used one, and we have not encountered statics. That being said, if this is something if it is a term that you need, then we can definitely add it. We didn't add it because we didn't need it, but it's something easy to add if it is needed.
I would say I've seen several studies that are static exposures where they just do it once and do it. The other thing that I noticed is that obviously the focus is on embryos, larvae, and perhaps there didn't seem to be an option to capture whether it was an acute or a chronic exposure. Obviously, there was a mention of what was the age of the organism at the start of exposure, what was the name of what was the age of the organism at the end of exposure. But thinking in terms of the reuse of data, having a filtering option where you can just download the data and filter to just those that are acute or chronic would be quite a beneficial addition, I would feel, to the end product. But that's just a suggestion.
Yes, this I was going to say, this is a great suggestion and a great comment. As I said, for the same reason that at this point, we didn't see, like, everything that we have seen in Zebrafish fits in this this is when you start, this is when you end the experiment, this is when you look at the phenotype. But definitely, if having that chronic versus acute is something that is helpful and needed, we will tweak the data model and make sure that it is represented. So thank you for the suggestion.
Thank you. Thank you for the work.
Okay, I see there is a question in the chat. Sabrina, can you see it, or would you like me to read? There is a question from Adrian Green.
I do not see any.
Okay, so
okay, that's that. Yeah, I had to. With data regarding the microscopy platform and image capture setting, be reporting with data. So this question is has to do with microscopy data information. So we right now we have minimal information that we're planning on adding to the image. So if I remember well, when somebody submit an image, they can also add information about I think the microscope and how the resolution, that's what I was looking for, this sort of information. Right now, this information is very, like, there's not a lot of information that we added in our data model, just because we need to have that discussion and we need to see what is needed for this. So long story short is there will be some information about microscopy. At this point, it's not detailed, but if it is something that is needed and something that especially other people need it, then definitely we can expand on it.
Yes, details of data collections are very important for data reproducibility.
Yes.
Yeah, great question. I see that Chris also added a question in the Q&A regarding have you encountered situation when there is a community disagreement on phenotype annotation, or perhaps phenotype is a bias or a combination of multiple phenotypes? If so, how did you approach that?
So we expect a lot of disagreement. We haven't. So we have so far we have really done a lot of exercises. It's nothing official because we're still working on the annotation tool. But we do know that there will be disagreement, and we do know that there will be combination of phenotypes. So the way we address the combination of phenotype is we allowing one image with multiple phenotype terms, because sometimes your image might show, just you have an image of the head and there might be like 5 different phenotypes. So we allow that. In terms of disagreement, this is where we really need to come up with an agreement and governance from the community. We know that we, being the ZAPP team, and people like me who is not an expert in phenotype annotation, we are not going to tell somebody you're correct, you're not correct. But we're thinking of ways for so we will accept whatever people are saying. We're talking of ways to have the community give the quote unquote peer review on that, the way we're thinking about right now. And again, it's a discussion, so it's not the final decision, but we're thinking of a way for people to somehow like agree or disagree with this information, like thumbs up, thumbs down, something like that. And then if there's, I don't know, like a lot of thumbs down, maybe, having a message to the submitter saying maybe you need to review your information, or as a user, if you see an image with a lot of and information with a lot of thumbs down, whether or not you want to trust this. So this is our idea of how to address the community disagreement. There might come moments where we want to have focused workshop and discussions about not only how we address this disagreement, but also how do we proceed if something is seemed to be very wrong or very, highly disagreeable. But in general, we want to make sure that everybody has a voice, and then we will make sure that people have a way to identify what they want to believe or not.
Yeah. Not an easy question to get to a consensus. I have another question, if I may. So regarding, it's a two-part question. So when you considered ontology terms and developed new ontology terms, one, did you identify any specific gaps in ontology? I don't want to say classes, but kind of types of terms that are missing. And when you add a new term, how do you consult with the community? How do you make sure that the term is agreed upon? It doesn't come only from your group, but agreed upon by multiple groups that work in the field.
Mm-hmm. I think I heard most of your questions. So if I'm not answering something, just tell me. In terms of lack of information in the ontology, definitely, like everything that was exposure route or exposure regimen, we couldn't find anything, and it really searching around, it looked like the exposure ontology was really where it was needed, and so we worked with that ontology to get those terms created. So that's really a place where we found a gap. I'm sure we will find some more gaps when we start, working with the community. But at this point, this is what we have found.
So where is this now? Is it EXO or EXO? Which ontology did you?
This one was EXO, so the exposure ontology. And so the way almost all those ontology works, I want to say all of them, but most of them, is that they have GitHub issues that is open to the public. So usually the way it works is that somebody from the community can go on the tracker. Everybody from the community can see what is being requested, and people can have the discussion on that GitHub issue to have those discussions if they agree, if they disagree, this sort of thing. So this is where the discussion with the community comes. I have to say, because I work a lot with ontology, if, let's say, we created those terms, they are now in the ontology, somebody disagree with it, we can go back and have those discussions and then tweak those terms. So the discussion with the community can either be on the GitHub issue itself or through the ontology, or sometimes what I expect is that some of those discussions will be at workshops. So for example, we are already planning on having workshops to discuss very specific phenotype, like brain phenotype or jaw phenotype. And then in this point, we will have the conversation with the community before we make the changes in the ontology.
Mm-hmm.
Was that all the questions? Okay.
Yes. Mm-hmm. Yes. Thank you very much. I'm afraid we are out of time, so I will take the opportunity to thank you for coming to this meeting and presenting your work. Our next webinar featuring environmental standards program will be on March 13 at 2 p.m. and the additional information will be sent via ELK listserv. Thank you very much, Sabrina. Thank you very much, everyone who joined us this Friday afternoon. Stay warm and safe. Be prepared to cold weather. Thank you.
Thank you very much. And if there's any questions, email me. I'm happy to discuss. So thank you for having me, and thank you everyone for your attention.